Welcome, folks. So, in our last couple of lectures, we've talked about how the body can engage potential pathogens and foreign agents by, by basically creating barriers and clearance in the innate immune system, but also specifically targeting and eliminating them in the adaptive immune system so that the host is not infected. But sometimes pathogens do get through the innate and adaptive immune system, and the host is infected. And so in chapter 10 and also in chapter 17, we talk more about what are kind of the artificial mechanisms or the chemical mechanisms that we can use to then address an infection. And specifically in chapter 10, we're looking at both natural and various levels of synthetic antimicrobials and antivirals. And then in chapter 17, we're talking about vaccines. Okay. Um, so a couple of notes before I have you review. There's this figure that's really going to guide our discussion today, 10.2 in the book, that goes through the different mechanisms of action that we use for antimicrobials and gives examples. Now, for the learning objectives, we're specifically just going to take a subset of these that are representative of the different mechanisms of action, and that's what we're going to be responsible for in terms of the learning objectives. So this image here is the first link in the description below that allows you maybe a really good kind of basis for your notes for the lecture today. So either pull it up or print it potentially, but I would just recommend having this image available to you. Basically, the notes that you add to that image or a table that you make that gives you similar information, that's going to be our discussion post for our D2L post for this chapter. So just keep track of that and you'll post that to D2L. Finally, our lecture today is really great parallel to the Ninja Nerd series on antibiotics. The Ninja Nerd series goes into more detail than we're responsible for in this class, but if it's something that you're interested in or that's going to be important to your career, I definitely recommend you going into the Ninja Nerd series as additional information and an augmentation of this lecture. So that's kind of our preface today, but I still recommend that you pause the video, look at previous content, preview chapter 10, and then come back into the lecture video. Okay, so we've already started to think of pathogens and their engagement with our body as sort of this little battle that's constantly, or many little battles that are constantly happening throughout our body. And we have these foot soldiers, which are basically our innate immune system that tries to prevent infection. And then we have these very specialized adaptive, the cells of our adaptive immune system that really target and take out pathogens. But every now and again, a pathogen will actually infect the host or our body. And in that way, we need to be considering, okay, what are some of the chemicals that we can use against that pathogen that can allow us to deal with that infection? Now, we don't really need to go far because in the world of biology, these little battles are happening all the time. Microorganisms in the environment, microorganisms in biofilms, microorganisms in different media are often competing for resources. And so there's a plethora of antimicrobial and antiviral chemicals out there that we can either pull from the environment or learn about from kind of environmental factors and then develop within the lab. And so we can learn from the little biological battles that are happening throughout ecosystems around us on how we can actually address the different battles or pathogens that are existing within our body as a host. And so, the first thing that you might think about if you're thinking about a pathogen and addressing that pathogen as an invader are what are the various components of that pathogen's existence that might be targeted by a different type of chemical or antimicrobial agent. And so, what we'll really focus on is breaking down the various chemicals that we use into mechanisms of action and thinking about different kind of examples of these mechanisms of action. So if we're talking about a bacterial pathogen, then we're going to think about, okay, what's holding this bacterium together? Okay, it's cell wall, it's cell membrane, it's cellular components. okay, maybe we can break down those structural components and in that way neutralize the bacterium. Or maybe we can look at its metabolism. What are the different types of metabolic activity, all the way down to the very basis of that activity in the DNA, and then all the way out to the actual implementation of that metabolic activity in proteins, and how we might address and actually attack that pathogen in those different ways and neutralize the pathogen. Now, if we're talking about viruses, viruses are, like we said, different from living agents or living pathogens. And so we'll be a little bit more thoughtful. We'll have unique, different types of chemicals that are antivirals that are separate from those that are antibacterials. Okay. Before we jump into that, let's talk about a little bit about vocabulary and history. Okay. So when we're looking at the various types of antibiotics or antimicrobials, various chemical agents that we might use to actually attack a pathogen, there are a number of different terms that are important to that discussion. So we have chemotherapeutic agents, which are just generally drugs that act against diseases or pathogens. And this can be as broad as various types of pharmaceuticals. But then we might be more specific and say, okay, what are antimicrobials specifically? And these are antibiotics. These are drugs that address infections that are caused by some sort of microbial agent. And then we can actually say, okay, well, in the natural world, there are antibiotics and antimicrobials that we can learn from and actually start to then synthesize various alterations or modifications onto those components that we can use within the pharmaceutical setting or the medical setting. And so these are called semi-synthetic antimicrobials or antibiotics. And then there are completely synthetic antibiotics or antimicrobials that are just completely lab generated. Okay. And so if we remember back to our very first chapters, we know that some of the first antibiotics that are known of in like recent medical history and recent microbiological history are penicillins. But if we remember back to that discussion and look back to history, we know that there are a number of antibiotics that were actually used predating Fleming's discovery. And so in terms of like the record of ancient Egyptians, like the embers of papyrus, which is basically a medical record of what ancient Egyptians were using, that dates back to 1550 BC. We have note of different types of molds that were being used as antibiotics. Also, in various skeletons that were kind of found in Nubian Africa that date back to like 350, 550 CE, and even late Roman time skeletons from Egypt. We actually see the traces of tetracycline in those skeletons. So for sure, folks were using antimicrobials and antibiotics before it became part of like Western medicine. But in terms of its initiation into Western medicine, we usually talk about Fleming and his kind of accidental discovery of penicillin because of molds on his bacterial lawns in his lab. Now, even once Fleming became aware of penicillin in the 1920s, there was still a delay before it was actually commercially available because up until till the 40s, we weren't really able to produce the antibiotic penicillin in enough quantities to actually even deliver to a human host. The first person actually to take penicillin as a human host was Albert Alexander, who was a soldier or a policeman who had a pretty bad infection throughout his body and particularly on his face. And actually, the dose that they gave him, they saw significant improvement in his case, but then they literally ran out of penicillin because it was so hard to extract. that Alexander or Albert Alexander actually didn't make it. He eventually, the infection came back and he died of that infection. And so there was significant amount of effort that was put into finding a strain of the Penicillium fungus that would actually have increased penicillin production. And so in the 40s, the US Department of Agriculture lab in Peoria, Illinois was tasked with finding the strain. And it's very interesting because they were actually having molds from across the world shipped into this lab, and they were checking the penicillin production of these molds. And eventually, the strain that became the Penicillium mold that we use to produce penicillin, and that probably had one of the most significant impacts on saving lives in the Second World War and then throughout history after that, was actually just found in the city of Peoria by one of the lab techs in the lab, who her goal was to just collect as many mold samples as possible. And she would collect them from local fruit stands. And eventually she collected this mold from a cantaloupe from a fruit stand in the city of Peoria. And when they tested it in the lab, that mold, for some reason, that strain produced about 200 more times penicillin than the strains that they had been working with previously. And so, what I'm saying with this history is we often think of antibiotics and the start of antibiotics as, Alexander Fleming's discovery, but knowing that that history is longer. And also, Alexander Fleming's wife as well was a big part of his lab, Amelia Fleming, and she did a lot of work with Staph aureus. And she also has just a really cool history with being kind of an amazing physician, an amazing microbiologist, but also a really, really important kind of part of resistance and uprising against kind of the Nazi regime of that time during, leading up to World War II. So just, amazing folks and amazing history all around the discovery of penicillin and all of the work that went into the immense life-saving kind of aspects of antibiotics. So just as a comparison, in World War I, about 1 in 5 soldiers actually died from bacterial pneumonia or infectious disease, and probably even more than that. But then in World War II, it was down to about 1 in 100. And a lot of that has to do with penicillin and kind of medical practices that were evolving at that time in terms of aseptic technique, so on and so forth. So, really understanding the importance of antibiotics, but also the history of antibiotics, is essential to this discussion. Now, we're going to talk more about specific antibiotics, but remember that many of them are coming from natural sources. And so, just like penicillin comes from a mold, there's a number of fungal organisms, bacterial organisms, and you can see the list here that are producing these antibiotics that we kind of base our understanding of antibiotics on, but then we can then go into the lab and add various components to them or synthesize similar chemicals. But we're learning from fungal organisms and bacterial organisms and organisms in the environment that are producing these chemicals. Okay. And so let's talk more about some of these. You see like penicillin, vancomycin, rifampin. We're going to talk about a number of these, but I want just to make one really important note as we begin our discussion of antimicrobials and vaccines. And that is a note about access. So, generally, throughout medicine, it is really important to note and to acknowledge the history of institutional injustice within medical systems. And that is particularly true of antibiotics and access to antibiotics, and vaccines and access to vaccines. And so, just acknowledging that these systems have really been biased towards an understanding that predominantly kind of gives us information around kind of Western scientists in a very biased way over these last couple 100 to 200 years of discovery, ignoring some of that really important indigenous knowledge, but then also that continues through in our application of these techniques and these medicines and tools. And so, really being aware and thoughtful of that and how that comes is kind of the basis of our medical system is really essential. Now, there are a couple of case studies in these resources that I'm giving you here. Everything is tuberculosis is really about access to medicine, less than being about tuberculosis, the disease itself. And this TED Talk here by Dr. Mary Bassett about racism in the medical system and the importance of medical activism to make changes. So I just want us to, take time to watch this TED Talk. So go ahead and pause the video, jump into the TED Talk, watch that. If you have time to read this book, it's not something you'll be tested on or required to read during this term, but I would recommend just thinking more about access in terms of the medical system and access to these antimicrobials and vaccines. We're also going to come back at the end of this chapter to a discussion of overuse of antimicrobials and antibiotics and how that is also a big part of a lot of, just issues that we're seeing across the board in the medical system. But stay tuned for that. Okay, so when we're talking about antimicrobials, there are chemicals that do different things. They act on the pathogen in different ways. Some of them are what we call bacteriostatic, which means they essentially stop some essential function within the bacteria without killing it. There are others called bactericidal, and these will essentially kill the pathogen on site. So you can kind of think of bactericidal as kind of like an execution sentence, whereas bacteriostatic might be more of like a life sentence in jail, where that pathogen is no longer able to replicate, reproduce, and continue its infection of the host, but it's still technically alive within the host. Also, we want to be thinking about the spectrum of activity, depending on the mechanism of action of these different types of chemicals. Some of them will be broad spectrum in that they will impact a number of different pathogens. But if it's so broad that it also impacts the host, then that can be a really big issue. Some will be narrow in their spectrum and will impact very specific organisms. And it's really important that we're thoughtful in how we actually prescribe those antibiotics so that they are specific to specific pathogens, or they might be limited in their impact to, specific types of organisms. Okay. And so this comes to a discussion of selective toxicity. We really want antimicrobials to be accessible. And we also want them to be selectively toxic. That means we want them to actually impact the pathogen of interest and not the host. And also, I'll add to that, we also don't want it to impact the microbiome of the host, which is an important part of the host and the host defenses. So, for example, you'll look at some of these different chemicals that we use and see that they target, for example, the peptidoglycan cell wall of bacterial pathogens, which we don't have peptidoglycan in our cells. So, as a host, a chemical that targeted the peptidoglycan cell wall of a pathogen would be less likely to negatively impact our cells. Okay, same for ribosomes. The structure of prokaryotic ribosomes and eukaryotic ribosomes is different in both subunits. And so when you have a chemical or an antimicrobial that targets ribosomes, then it's a little bit more selective because it's less likely to impact, although there's still sometimes some overlap, but it's less likely to impact the host ribosomes since our ribosomes are structurally different from prokaryotic ribosomes. Okay. And we mentioned spectrum of action, narrow and broad. And we mentioned selective toxicity. So let's go ahead and talk a little bit about the modes of action of different types of antimicrobials. This will give us an overview of how you might kind of take out, essentially, a pathogen by targeting different aspects of that pathogen.
Chemotherapeutic agents are antimicrobial drugs that fight disease by targeting a particular structural difference between human cells and those of the microbe. Because bacteria have many such differences, it's much easier to develop antibacterial drugs. Viruses depend on the host cell's machinery, so there are fewer differences to target. And eukaryotic pathogens, such as fungi and protozoa, have fewer differences from eukaryotic human cells, making it difficult to find drugs that have selective toxicity against these pathogens. Antibacterial drugs have several different modes of action. One of the ways in which antibacterial drugs can kill bacterial cells is to block the formation of bacterial cell walls. Normally, the peptidoglycan layers are held together with peptide crosslinks. Penicillin and its relatives block the formation of these peptide crosslinks. This weakens the cell wall, ultimately causing the cell to lyse. Antibacterial drugs such as polymyxin attach to phospholipids in the bacterial cytoplasmic membrane and interfere with its integrity. Eventually, the cell lyses. Because this mode of action also interferes with human cell membranes, this antibiotic is used only on the skin, where the outer layers of cells are already dead. Antibacterial drugs such as tetracycline, chloramphenicol, and streptomycin kill bacteria by inhibiting protein synthesis. Tetracyclines block the docking site of the transfer RNA, preventing amino acids from being added. Chloramphenicol prevents formation of peptide bonds between amino acids. Macrolides like erythromycin block the movement of messenger RNA through the ribosome. Aminoglycosides like streptomycin cause a change in the shape of the smaller 30S ribosome subunit, resulting in misreading of messenger RNA and insertion of incorrect amino acids. Bacteria have certain metabolic pathways that are not found in humans. One of these is the pathway for synthesis of folic acid, a coenzyme used in the synthesis of nucleic acids. Humans do not have such a pathway and require folic acid as a dietary supplement. One of the substrates in this pathway is para-aminobenzoic acid, or PABA. Antibiotics such as sulfonamides or sulfa drugs block a step in the folic acid synthesis pathway by acting as a competitive inhibitor to PABA. This prevents synthesis of folic acid and therefore synthesis of nucleic acids. The bacterium is unable to reproduce. Most organisms synthesize nucleic acids in a very similar manner, so it's much more difficult to find differences to target for these pathways. However, there is a group of synthetic drugs called quinolones and fluoroquinolones that block DNA gyrase in bacterial DNA. They have little effect on eukaryotic cells. Other drugs, such as rifampin, bind more readily to RNA polymerase in prokaryotes than eukaryotes and can be used to block transcription.
Okay, so let's just go through these kind of mechanisms of action one by one and learn a little bit more about some of the representative antibiotics within each group. Okay, I will note that I've reorganized the series in which we're introduced to antibiotics to match a little bit more logically how you would kind of envision a cell. So first we'll go from the outside of the cell, so the cell wall, to the cell membrane, and then we'll go into the cell, starting with kind of the central dogma as our guide, back to DNA, inhibition of DNA replication, and then RNA protein. And so in that way, we can just kind of go through them in a way that's a little bit more logical. Okay, so let's start with the outside of the cell. We're going to talk about antibiotics that inhibit cell wall synthesis. Okay, so pathogen, particularly one that's bacterial, and that's going to be our focus for the first part of this class, our class today. At the end, we'll talk about some antivirals. I'm sorry. So pathogens that are bacterial, they're going to have that peptidoglycan either fully on the outside of the cell, or for our gram negatives, they're going to at least have a thin peptidoglycan layer internal to the outermost membrane. So, there are a number of antibiotics that will specifically target cell wall synthesis. And one of these groups you're probably very familiar with. These are the beta-lactams, which include penicillins and their relatives. Essentially, what's happening is the beta-lactams, and the beta-lactams have a specific structure. They have like a ring unit that allows them to bind to enzymes that are important in actually building the links between the layers of the peptidoglycan. If you remember back, peptidoglycan is made out of NAG/NAM subunits that are connected, but they're also cross-linked. All of those layers are cross-linked. And so if you can disrupt those linkages, then you can basically allow for that cell wall to fall apart. Okay? And so um, like we said, there is a number of like natural antibiotics like penicillins that will do this. There are also some semi-synthetic derivatives of penicillins, which we basically altered a little bit to make more effective against cell wall synthesis. Okay. So if you're looking at particularly gram-positive organisms, then you're going to see that their cell envelope has a lipid bilayer. This is just a reminder because we've seen this before in the class. And then you can see that peptidoglycan layer. These beads that are turquoise and blue, those are our NAG and NAM subunits. And then those layers are held together by cross linkages. And those cross linkages are really important to holding together the whole peptidoglycan layer or cell wall, or the cell wall with the peptidoglycan layers. And so when you have anything that would disrupt that integrity, then that's essentially going to puncture that outer wall of the cell, and that's going to lead the cell just to explode and die. Okay. So our beta lactams, and including penicillin, they have this beta lactam ring that is conserved within the different groups or the different types of penicillins. And that ring allows them to essentially engage with an enzyme that connects the cross linkages within the peptidoglycan cell wall. So if we're going to go back to this image here, these purple cross linkages, okay? And this is an enzyme called transpeptidase, and it essentially just makes those connections. So as the bacteria is growing and reproducing, this transpeptidase just connects those links between the layers of the peptidoglycan cell wall. And that's how the cell grows. That's how the cell kind of maintains its integrity over time. But if and it stays connected, right? Which you see here. But if you have your beta lactam come in and engage with this transpeptidase and actually stop it from its action, then you're going to end up with layers of NAG and NAM peptidoglycan layers that are not connected to one another. And so what I imagine is kind of like if you're making a layered cake, if the temperature or the composition of the icing that you use or the cream that you use to hold those layers together is wrong, then actually the cake will just start to slide apart and the layers will not be connected well, and that whole cake or the integrity of that cake will fall apart. And so that's essentially what happens to cells that are exposed to penicillin is because this transpeptidase is no longer functional, is inhibited, then the layers of the cell wall are no longer held together, and the integrity falls apart of the whole cellular envelope. Okay, so that's the example of penicillin and beta lactams generally. Penicillin is an example within the class of beta lactams. For example, we use ampicillin in the lab. That is a relative of penicillin. That's also a beta lactam. Okay. Then you have other ways of disrupting the integrity of the cell wall. So we have lipoglycopeptides. That's a group of antibiotics, and an example is vancomycin, which you might be familiar with. They interfere with the peptidoglycan cell wall in just like a slightly different way. And in that they actually inhibit the connection between the NAG and NAM subunits in the first place. And so vancomycin is actually inhibiting the production of the cell wall at all. Whereas penicillins, once the layers of the cell wall are produced, they're not linked together. So this is actually vancomycin and lipoglycopeptides are actually inhibiting an earlier step in the formation of the cell wall than our beta lactams. And so if we go back to our transpeptidase here, actually this is going back to our beta lactams, but I think I don't have an image specifically for vancomycin, but you can imagine that integrity between those NAG and NAM subunits, they're just not able to come together in the first place, and so you don't have the connection of those subunits within the cell wall. And so that's our beta lactams and our vancomycin. Both of those are impacting the cell wall of the pathogen. Okay. Um, so when you're breaking apart that cell wall, that is considered what we said bactericidal versus bacteriostatic. When the cell wall's integrity is actually disrupted and basically the cell just like leaks through its own cell wall, that's going to kill the cell. So those tend to be bactericidal. Okay. We have a couple other antibiotics that also inhibit the cell wall synthesis. So bacitracin is an example that bacitracin actually inhibits the movement of NAG and NAM subunits even to the surface of the cell. And so that's an even earlier step in the formation of the cell wall that's inhibited. And in that way, the cell wall is never even made in the first place. And so that is another example of an antibiotic that inhibits cell wall synthesis. Okay. I think we mentioned that these tend to be effective against gram-positive organisms, and they prevent the organism from maintaining its own integrity, and it tends to be bactericidal. And there are also some antibiotics that work against fungal cell walls, but those are going to be a little bit different because the structure of fungal cell walls is a little bit different within those eukaryotic pathogens as well. So first we talked about chemicals or antimicrobials that might inhibit the production or the synthesis in various ways of the cell wall. And we gave some examples of penicillin, lipoglycopeptides, and bacitracin. Now let's talk a little bit more. Like I said, I rearranged some of these. We're going to move into the disruptions of the cytoplasmic membrane or the cell membrane. And so the cell wall, right under that is the cell membrane. There are chemicals that will specifically target the cell membrane, and that's how they will disrupt the activity of our pathogen. Okay. So there are a number of examples. We're going to focus on just polymyxins. So if you remember, the cell membrane is a phospholipid bilayer that's active. It has a number of transmembrane proteins, and it's allowing for the cell to kind of maintain its engagement with the environment, but its integrity from the environment, right? And so essentially, with your polymyxins, they actually disrupt the plasma membrane. They're particularly effective against gram-negative organisms because their outermost layer is a phospholipid bilayer rather than a peptidoglycan cell wall. So we said our penicillins and so on and so forth, they're targeting that cell wall that's on the outside of potentially like a gram positive, although they can sometimes be effective against gram negatives as well. But for our polymyxin, specifically targeting a cell membrane and tends to be more effective against gram negative organisms. But also, since we have cell membranes on our cells as well, there's less selective toxicity because it can actually impact our cells as well. And so there are some side effects or potential negative side effects from using some of these antibiotics as well. So polymyxin, what does it do? Polymyxin essentially its structure is similar in a way to like a surfactant or even kind of mimics some of the molecules within the cell wall in that it can insert itself because it has fatty acid chain, but also it has positive charges as well. It can literally insert itself into the membrane and just create little punctures or little holes within the membrane. And then that literally just, the cell is no longer able to maintain its own integrity and it leaks out of its own cell membrane. And so it literally just here's a fatty acid chain, engages with the membrane, and because of the charges as well, because it has positive charges as well, it inserts itself, but it's so large that it just leads to leaking in the cell membrane. And in that way, it also kills the cell. Okay. This, like we said, tends to be effective against our gram-negative organisms. So specifically, Pseudomonas organisms and also some of your Enterobacteriaceae like E. coli, some of the organisms like H. influenzae, and it allows for targeting many of these, but it has less coverage against gram-positives. There are similar types of antibiotics that target eukaryotic infections, like fungal infections, but they're different because those cells are structured a little bit differently. But essentially, what they do is the same kind of action in that they make a pore within the membrane of the fungus, and actually, that is what disrupts the integrity of the cell. And again, these antimicrobials or antifungals that are against eukaryotic cells are going to have less selective toxicity against a human host than something like a polymyxin, which is against bacterial infection. Okay. So we're gonna go ahead and pause this part here, and I'm gonna let you review. We've talked so far about inhibition of pathogens by targeting the cell wall and the cell membrane, and then we're gonna move into a discussion of targeting of nucleic acid synthesis and also various metabolic pathways. But let's just go ahead and pause here, review, add to our diagram, and then we'll jump into part 2 of the lecture. Okay, see you there.